I'm Semi, and this is the endomembrane system. First up, we have the formation and pathway of a secretory protein. The formation of a secretory protein begins in the nucleus. The nucleolus is a region of the nucleus that contains DNA, RNA, and proteins. Other parts of the nucleus include the nuclear envelope and the nuclear pores. The nucleolus is the site of transcription, or the synthesis of mRNA using a DNA template. mRNA leaves the nucleus through the pores in the nuclear envelope. mRNA then arrives at the ribosomes on the surface of the rough endoplasmic reticulum. The ribosome translates the genetic code in the mRNA into an amino acid sequence. This results in the formation of a polypeptide. The polypeptide chain then enters the ER lumen where it folds into its appropriate shape. Once the protein has been formed, it departs from the ER in a transport vesicle. Next, the transport vesicle holding the protein arrives at the Golgi apparatus. The transport vesicle fuses with the cis phase of the Golgi apparatus to transfer its cargo into the lumen. As it moves from the cis phase to the trans phase, the protein undergoes various modifications. This includes the addition of external molecules to recognize the cell membrane. Finally, the protein emerges from the trans phase in a vesicle, which travels to the plasma membrane for secretion. Next is the formation and pathway of a lysosomal enzyme. A lysosomal enzyme is formed in a similar process as the secretory protein. mRNA leaves the nucleus. A polypeptide chain is created by the rough ER's ribosomes, and the enzyme is folded into its appropriate shape in the lumen. The enzyme then departs from the ER in a transport vesicle. The lysosomal enzyme travels from the cis phase of the Golgi apparatus to the trans phase. They get marked with the M6P mannose-6-phosphate group to recognize a lysosome. These enzymes depart from the trans phase and arrive at a lysosome in the cell. Otherwise, lysosomes can bud directly from the Golgi's trans phase, already having contained a lysosomal enzyme. Next, we have the formation and pathway of a misfolded secretory protein that is destroyed by the cell. A secretory protein is formed when mRNA travels from the nucleus to the rough ER's ribosome. Misfolded proteins are often due to mutations in the polypeptide sequence during transcription or translation. These mutations can cause incorrect folding of the protein in the rough ER lumen. The first way that misfolded proteins can be destroyed is through ER-associated degradation. In ERAD, the misfolded protein leaves the ER and is destroyed by a proteasome. A proteasome is a machine that breaks up a protein into individual amino acids. The second way a protein can be destroyed is by lysosomal degradation. During this process, a misfolded protein leaves the ER in a vesicle. The vesicle fuses with the lysosome, and the lysosome's hydrolytic enzymes break down the protein. Finally, we have the formation and pathway of a glycoprotein displayed on the outside of the cell. For the formation of a glycoprotein, DNA will first undergo transcription into mRNA. mRNA will leave the nucleus, and a polypeptide will be created by the rough ER's ribosomes. Then the enzyme will be folded into its appropriate shape in the lumen. Carbohydrates are then covalently attached to the protein by the ER membrane's enzymes. This results in the formation of a glycoprotein in the lumen. The glycoproteins then depart from the ER in transport vesicles. Once they are carried and deposited into the Golgi apparatus, Glycoproteins will move from the cis phase to the trans phase where a vesicle buds. The transport vesicles carry the proteins to the cell membrane and release them to the extracellular space. Glycoproteins serve as antigen receptors on the surface of the cell.